Well, our first presenter today is Vincent Marion, who's the CEO of ALMS Therapeutics, or ALMS Therapeutics. So, Vincent, are you with us?
Yes, I am.
Excellent. Well, if you're able to present your screen, Vincent, we'll hand over to you and we'll see you on the flip side in 12 minutes. Good luck.
So I'm going to talk today about ALMS Therapeutics and mainly about PATAS, which is a new molecular entity to treat type 2 diabetes and associated comorbidities. I'm the ALMS founder, president, and acting CEO, but I'm also an academic and director of Laboratory of Medical Genetics here in Strasbourg. So next slide, please. So we are a privately held biotech company. We are preclinical stage, and we are specializing in peptide therapy. We were founded in 2017, and we are based in Strasbourg, France. So what we do is we harness rare genetic disorders, disease-derived knowledge to treat type 2 diabetes and associated comorbidities. And after several years of research, our lead compound, we term PATAS, is a staple peptide targeting a unique protein-protein interaction of ALMS1, a protein identified in an ultra-rare genetic disorder characterized by early onset and severe type 2 diabetes associated with non-alcoholic fatty liver disease and fibrosis. Next one, please. So our strategy is always to start from the clinic. So we have a patient-based discovery platform. We develop therapeutic options using relevant biology by trying to really understand the signaling pathway behind this disease. And of course, we try to expand that to disease model and extensive safety studies using and basing ourselves on strong human genetic data, as you will see. And of course, we have to protect that. So we have an extensive patent portfolio. And in the next slide, that is to show you our starting point, which is really academic research on a rare genetic disorder group called the ciliopathies. And on the left side is kind of how we operate. So we get the patients in the clinic, we get to have what we call the clinical research protocol. And our starting point is really looking at the patients. So once we find a group with clinical disorders, we try to molecular to do identification on a basically using high throughput sequencing like exome and genome sequencing. And then once we find the causative genes, we really characterize that. And then using basic and applied research, we try to decipher the signaling pathway linking these causative genes with these complex disorders. And this is how we basically design targeted therapeutic options that will eventually lead us to go back to the patients, not only for the rare genetic disorders, but then expand that to the common disorders. And on the right side here is shown the disease basket of these ciliopathies. On the x-axis here are basically the different syndromes that we have. And on the y-axis are the clinical cardinal features that define these disorders. And one of them being obesity associated with type 2 diabetes, for instance, like Alström syndrome, but also the Bardet-Biedl syndrome. So next one, please. So the Alström syndrome and its connection between the adipocyte driving type 2 connection. The ciliopathy Alström syndrome is really characterized by very early onset type 2 diabetes with severe insulin resistance. Basically, by the age of 5 years old, these kids will be severely type 2 diabetic. And what we're seeing is that they also develop progressive hepatic failure, which is a form of accelerated non-alcoholic fatty liver disease. And the interesting thing about this syndrome is that it is a monogenic disease in the sense that it has only one gene, called the ALMS1 gene, which basically gave the name to the company. And with this one gene, we have a very strong starting point. So one unique starting point with leading to such a complex phenotype like associated type 2 diabetes and non-alcoholic fatty liver disease. And this gene codes for a massive protein called ALMS1 gene, a protein. And this massive protein has never been crystallized. So we had to use also in silico, which everyone pretty much do right now, is to really assist us in trying to identify how ALMS1 protein were related to these different phenotypes. So next one. So our starting point, like we said, is the clinic. So Alström syndrome, we just published, came out in September of this year. The relative adipose tissue failure in Alström syndrome drives the obesity-induced insulin resistance. So starting from the patient data on the left side, what we've been doing is by doing microdialysis in the patients, so we were able to show that the glycerol excretion was much higher compared to BMI match group controls. So in other way, they had a higher glycerol excretion in the arms compared to the obese, normal obese. And also we had a pyruvate lactate ratio, which was much higher compared to BMI. And pyruvate lactate ratio translates to mitochondrial dysfunction. So these patients were suffering from very severe and specific adipocytes dysfunction. And when we had biopsies from these patients, again compared to BMI match control, as you see below on the left side, you see that the cells of the ALMS patients were much bigger. So this is hypertrophic phenotype is really characteristic of a disease adipocyte. And then switching from the human data to, oops, no, too fast, to the mouse data, what we did was to use a naturally occurring mutant mouse from Australia called the fatty Aussie mouse, which recapitulated all the syndromic conditions like the glycemia. And what we did after that, we infused radioactive glucose to see what was the tissue which was driving the type 2 phenotype. And that's bottom right. And what you see here is that in the ALMS mouse, knockout mouse, it was a very specific repression of the adipose tissue absorption, which was driving the whole phenotype. The liver, heart, gastrocnemic muscle, and even the brain was unaffected. It was really the adipocyte which was impaired in ALMS knockout mouse. Next one, please. So then what we did was to really go for in vitro. So we knocked down ALMS in vitro. So we saw that we had a cellular glucose absorption, which was repressed under insulin action. That's on the left side. And when we look at the mechanism on the right side, for the control, without insulin and plus insulin in the middle, you see that these little invaginations, which corresponds to GLUT4 getting to the plasma membrane for glucose absorption, was there in the controls, but were not present when you were inactivating ALMS1 and giving insulin. In other words, if you don't have ALMS1, the GLUT4 cannot come to your membrane under insulin and hence absorb glucose. So that's why these adipocytes could not absorb glucose. Next one, please. So long story short, and how we got to the PATAS origin basically as a novel biology to treat type 2 diabetes and non-alcoholic fatty liver disease, is that on the right side, you see a scheme that we've been able to show that under insulin action, you have a classical insulin signaling pathway coming to what we call ALMS1, releasing alpha-PKC to activate proton pumps. And that proton pump will then allow the GLUT4 sorting vesicles to go to the membrane and fuse with the plasma membrane, which will lead to GLUT4 in the plasma membrane to absorb glucose inside. So alpha-PKC is released from ALMS1 in response to insulin in the adipocyte, and alpha-PKC activates this GLUT4 fusion with the plasma membrane. So PATAS approach was that if you're insulin resistant, we would trigger insulin-independent glucose absorption by releasing alpha-PKC from ALMS1 in the adipocyte. Next one, please. So what we got from our all basically experiments of nature and the human genetics taught us is that when you have insulin and you're insulin resistant and alpha-PKC remains bound to ALMS1, the adipocyte can no longer absorb glucose, which has a high circulation level. But instead of absorbing glucose, since it is a living cell, it switches from glucose absorption to lipid absorption by upregulating FAT-P2. And this upregulation of FAT-P2 associated with lipid absorption basically leads the cell to secrete very deleterious lipids that will then eventually go throughout the body, drive type 2 diabetes, non-alcoholic fatty liver disease, fibrosis, and metabolic syndrome at large. And the next slide, please. The PATAS method of action was really to think, okay, so if alpha-PKC is not being released from ALMS 1 due to insulin resistance, we would develop and design a peptide that will really go and target alpha-PKC and ALMS 1 interaction, and thereby release alpha-PKC from ALMS 1, activate proton pumps that will lead to GLUT4 going to the membrane. Once you get this fusion with GLUT4 on the membrane, you get glucose absorption. Glucose absorptions in the cell will lead to the restoration of a physiological energy source of the adipocyte, which is glucose instead of the lipids. So getting back access to glucose means that the adipocyte no longer need to eat the lipids, so it starts repressing its lipid absorption. And hence, with glucose restoration, the adipocyte starts secreting very healthy lipids. And these healthy lipids will then eventually treat type 2 diabetes, improve liver function, fibrosis, and also improve metabolic syndrome at large. And this is how PATAS works, by restoring glucose absorption in a diseased adipocyte. Next one, please. So, of course, the relationship between adipose tissue and non-alcoholic fatty liver disease, as you know, a lot of stories are coming around with studies on the liver with NASH, non-alcoholic fatty liver disease. But what is very important to keep in mind is, so you have a lot of lipids in the liver when you get this non-alcoholic fatty liver disease, but where do they originate from? And there was a very interesting paper which I think everyone trying to cure the liver should look into, was published in JCI by this group here. Sources of fatty acids stored in liver and secreted via, and this shows that it originates from the adipocyte. So what we know, 2/3 of a hepatic triglyceride originate from the adipose tissue. So basically, if you want to treat and cure the liver, you better start by treating the adipose tissue, which secretes 2/3 of what you will end up having in the liver. And that's PATAS what it does. It treats the adipose tissue and hence cure the liver. Next one. So what we've been able to show is that using different mouse models, we are showing primarily mouse data, but we have mouse, rats, and dogs data, showing that if you would go with a weekly injection of PATAS, a subcutaneous administration, what we have been able to show is that we not only improve glucose intolerance in this diabetic mouse model of DBDB, but we also improve incretin levels. So biosynthesis and also endogenous incretin levels are maintained. And as shown here, GLP-1 stays quite to a normal level compared to vehicle-treated diabetic mice.
One minute, Vincent.
Yeah. We went to the most severe mouse model of NASH, a Japanese CRO called SMC, which had very severe liver steatosis. So we were able to decrease that by 60%, but also we were able to decrease fibrotic lesions with ballooning by more than 40%. And that was in their mouse model. Also in diet-induced obese mouse model, we saw that on electron microscopy, these fibrotic lesions were decreased, almost gone if we would keep the treatment long. So we also found that we were able to improve kidney function in the DBDB mice, but also, and very interestingly, we found that we were improving pancreatic function, and that by reducing the plaque. So when you're diabetic, you start having this plaque forming around the beta cells. We reduce these plaque, thereby allowing physical secretion of insulin to be reestablished. So that's why we were able to maintain pancreatic function, but also improve insulin secretion. And last but not least, we found that we were improving general fibrosis. And when we started looking at the lungs in these treated mice, we saw that we were decreasing the fibrotic lesions on electron microscopy in the DBDB mice, the diabetic mice. So next one. So as a summary, So PATAS is a new molecular entity. It triggers glucose entry in the adipocyte and restores natural cellular pheotropism in the adipocyte. By restoring physiological energy usage in the adipose tissue, this is associated with whole body benefits. PATAS shows activity as an anti-diabetic agent, GLP-1 secretagogue, anti-non-alcoholic fatty liver disease, fibrotic agents, improve pancreatic function with insulin secretion, and of course, improve kidney function. It is rapidly clear from the body, and it is effective at very low dosage. Next one. So PATAS characteristic and roadmap, it is a 25 amino acid, as I just said. It triggers glucose-independent insulin and absorption. And the interesting thing, it doesn't need complex formulation. It is just soluble in saline. So on the formulation, that's good. It shows beneficial systemic effects in preclinical proof of concept in different rodents and dogs, and we are planning to hopefully get into a phase 1 in the US by next year. So with that last slide, I would like to thank you, and I think that's the next slide. Thank you.
That's great, Vincent. Thank you. Well, very best of luck with your phase 1 next year. Can I just ask you before we move on to the next presenter, with your business, how have you actually been able to fund what you're doing up till now?
So we are basically, we've been raising money, obviously with VCs, the classical one. In France, when you're a French-based company, you get substantial amounts of non-dilutive money from the French state, which allows you really to drive up to pretty much the clinical phase. So a lot of help from the French state, and of course, independent VCs.
Great. Well, thanks very much, Vincent, and good luck with your business and your trials next year.